Tonight, your speaker is Dr. Sarah Wyatt, professor of environmental and plant biology, and the vice president of the local chapter of Sigma Xi. All right? For faculty around, Sigma Xi is going to start taking recommendations for new members, student members, so please think about that. Think about who you have that would be eligible and ready to join Sigma Xi. Sarah's topic for tonight is grounded in gravity. So, let's kill this.
Where's Sarah?
Go away. Is there anyone in this room that doesn't realize I'm Sarah? See? Okay. So, I'm going to leave it here for a minute because I didn't put a blank slide in there. The first thing I want to do tonight is think about plants and how plants respond to gravity. Most of you probably have some vague idea of gravity and how plants may respond, right? Shoots go up, roots go down. Fairly simplistic idea, right? But that's not always true. And if you look at some of the plants I brought today, definitely shoots are going up, roots are going down. When you germinate a seed, the seed knows which way to go, the shoots and roots, without you having to plant it right side up. Never thought about that, huh? You don't have to plant seeds right side up. Gravity tells it which way to go. Every once in a while somebody will say, oh no, it's the light. Really? It's underground. It's gravity, all right? So, one of the things you'll notice if you look around, we have a lot of plants here. Some of these guys have some stems going up. Some stems, as they get older, curve down, but the tips are still going up. You'll notice this in a lot of hanging basket types of plants. What happens is the stems want to go up, but often it's a weakness in the cell walls that cause it to drop over. It's just not strong enough to hold its own weight. So this is important to understand. There's a lot of different ways you can get plants that are or look like they're defective in a gravity response. This could just be they're not strong enough and they fall over. It could be that they actually have a major defect in gravity. So all of these things are important to think about when we start thinking about how we're going to tease this apart. You will also notice that I brought a couple of DNA molecules. We're going to interlace the two because most of my lab works with DNA. Several of my students are here. They're in the NASA t-shirts or the NSF t-shirts, the two current funding agencies we're working with. And they've been practicing their elevator speeches. So if you guys want to ask them questions, do so. We'll see how good they got. They were warned to. So we have all these plants that have very different responses to gravity. So you can't really talk about positive gravitropism or negative gravitropism. What you have to talk about is the gravitropic set point angle. Even a plant like this, that for the most part the stem is upright, the leaves and the side branches come off at angles. They're very specific angles, and they're also determined by gravity. So when you see a tree, and the tree is mostly upright, but you have branches, what you wind up with is each one of those branches has a gravity set point. If you remove this, I had this happen to me once with one of my students. This is my plant. This is our plant of choice. Some of you have them on your table. They're Arabidopsis, all right? This is Arabidopsis. And we do several experiments with it, and we were decapitating them and using the tops for a gravity response. Well, one of my students thought she'd be really, really bright, and it had lots of long stems, so she thought she could cut her work down, and she took all the side stems off as well and put them all on the plate as if they were even. Then rotated them. And, of course, the main stem came back up, and all the others came to whatever angle they were, and her data looked really, really funny until I figured out what she had done. Something that simple, the whole idea that each one of these has a different set point angle. Now, we use gravity in a lot of ways. A lot of the plants that we have, our agricultural plants, our horticultural plants, have been selected over the years, the thousands of years, because of their response to gravity. Think about corn. It would be really hard to harvest corn if it was laying on the ground. So these things that are very upright, you'll notice most of the crop species are very upright, so that we can go through and get them with the combine. Horticulturally, how many of you guys or your parents have a weeping something in your yard? Weeping somethings. Cherry tree, willow tree. These are artificially made. They find these really nice trees that have this really kind of a weeping character to them, not exactly going up. Now, the problem with this is the mutation that is causing the shoot of this plant to go down would also affect the roots potentially. If it affects the roots, you can imagine the reverse of this. All the roots are coming up. Well, if the roots are coming up, they're not really doing their job. Their job's to hold it in the ground and to get water and nutrients out of the soil, water and minerals, right? So what you'll see on these horticultural plants is a graft point. This little knot right here is a graft point where they've taken this gravity defective plant and grafted it to a rootstock that actually is normal for gravity response. That way the roots go down and the shoots do whatever this is the shoots are doing. So check your weeping stuff. When you go out and you buy these weeping plants, they've all been done this way. Because the defects, when you find something this unusual, the defects are quite remarkable. Okay? So I'm hoping that you guys will look at the plants around you maybe a little differently. So we'll put that one back there. So I've been interested in gravity and gravity signaling for, I don't know, 15 years or more. Now, the problem is, how do you actually do research on gravity? Gravity is all over Earth. What's the normal control? With gravity, without gravity? But we don't have without gravity. NASA doesn't have a no gravity room. So you're stuck with gravity. So how do we look at gravity signaling, which is what I'm most interested in? And there are several ways that we do this. The majority of my lab uses gravitropism. So we take our plants that are growing perfectly normally and we put them on their side and wait for them to bend. Several of you have plants on your tables that have been turned over and are working at gravity response. By the time I'm finished, you should see that they've come up, or at least come up to some extent. All right, especially with this plant and the little mints that we have here. These are mints. Mints do this really, really well. Throw a few of them out here. And of course, our vertical controls. So these are our treatment. They're on their side. These are our vertical controls. People have been doing this work for a long time. Darwin noticed gravity. Darwin talked about gravity. So when I got into this field, this was sort of the state of the art. Anytime you have a signaling event, it starts with a perception. So here we have a stimulus perception. It perceives gravity. Then there are several events that occur, and ultimately there's a response. For gravity, that's a differential growth. Now, just as I was getting into this field, people were figuring this piece out. This is auxin. It's a plant hormone. And it actually causes the differential growth that causes gravitropism. The problem is this piece. Here we have a biophysical stimulus. There are starch grains in specific cells in the plant, and when the plant falls over, those starch grains move. But when the starch grains move, then they cause something to happen. They start a signaling pathway. Just as if I touched you, there's a signaling pathway that runs to your brain and says you've been touched. Okay? So they're standing up, they're falling over. Now, this may not be the only thing. We actually know now that just statilus moving is not the only trigger. We have plants that don't have starch at all, and they still bend somewhat. They just don't bend as well or as fast. However, the principle is still the same. It is biophysical. We're taking this biophysical, or a plant is, taking this biophysical stimulus and turning it into a biochemical event. A biochemical event. Plants growing perfectly normally, then auxin gets shifted to one side or the other, and all of a sudden that side starts to grow, and you get curvature. Okay? Now, what I'm interested in are these links. What links these things together? How do you turn something that's biophysical into something that's biochemical? I got to keep an eye on the clock. Okay. So what I did was I designed a mutant screen. Looking at mutants is a great way to find the genes that you're looking for, no matter what the process. So yeah, so there you have a black box. This is my favorite cartoon. You see the guys talking to each other, lots of on one end, lots of equations on the other. And of course, right in the middle it says, and then a miracle occurs. Which pretty much is the way it goes. And then of course the caption, I think you should be more explicit here in step 2. So we're going to try to be a little more explicit here in step 2. So what I did, there were lots of mutants. All the little things you saw on this slide, all these little things in here, all of these are mutants. These are all names of mutants. So when I was thinking about designing my mutant screen, I didn't want to pick up all the mutants everybody else does. It's a growth defect. So any plant that's a dwarf or has some growth defect is going to be, by definition, agravitropic. So it's going to mess up its gravity response. So what I did was I used a cold treatment. You take these plants, you put them, they're vertical, you put them on their side at 4 degrees in your refrigerator. Put them in your fridge. They don't bend. Physiology has a real hard time at 4 degrees. It's really slow. So most of the biochemistry isn't going to happen. Then you bring them out of the cold and you sit them vertically. And they bend. They remember the signal. The animal biologists don't like me call it memory because it's not the same mechanism. So the mutants are actually called gravity persistent signal. There's some signal that gets turned on in the cold. The statilus fall. The signal gets turned on. You bring it out. You put it up. And all of a sudden it's released. And now it allows for bending. Does that make sense? So I used that as my mutant screen, as complex as it is. So this is basically it. The other cool part about this, and I love this piece, is if you put them in the cold on their side, and then you stand them vertically in the cold, and then bring them out, they forget. So there's a time limit there where they actually forget the stimulus. So that signal dissipates over time. Okay. So I'm not going to spend a whole lot of time on this, but welcome to the gypsies. These are the mutants that I isolated. If the plant were normal, this is wild type, this is what it should do. It should bend that way after this stimulus. If you notice, Gypsy 1 hasn't got a clue. It's completely forgotten everything. It doesn't know it even had a stimulus. Even more curious is Gypsy 2. Did I say? Yeah, Gypsy 2. Gypsy 2 bends exactly the wrong way. Before this mutant, we never had a wrong way mutant. We had lazy ones that couldn't stand up, but we never had one that intentionally went the wrong direction. And this was something there was no way I could have predicted. I was expecting all my mutants. I was expecting to get Gypsy 1, and that was it. They just didn't remember. But not only did I get those two, I also got Gypsy 3. It goes the right direction, but it never stops. It just keeps on going, and occasionally it'll go all the way around in a curlicue. Okay? Really kind of interesting mutants. Fun to play with. I couldn't bring them to show you the real plants because they're a little finicky and it's really cold outside. But that's Gypsy 1, 2, and 3. And then later we found others. Gypsy 4 is similar to 1, kind of no response. We have another over-responder. I call it the overachiever. Then you have Gypsy 6. Gypsy 6 eluded us for a while because what Gypsy 6 does, it's in the cold, it comes out, it's supposed to go this way. Gypsy 2 goes that way, and it's supposed to go this way. Gypsy 6 goes this way, out of the plane of the camera, right? So it points right at the camera because we're shooting everything from the side, and it points right to us or directly away from us. So we couldn't get a picture of it forever. We couldn't figure out what was going on forever. So then we put the camera on top of it so we could actually get the curvature and the direction it was going. So that's Gypsy 6. It's another wrong way. It's just not exactly wrong way. So it does a kind of a 90-degree. And Gypsy 7 is just slow. We actually discovered that one by accident. One of my students had them. They were doing a treatment. They were doing a Gypsy treatment. And they forgot about it. And they went back later. And it should have all been over. But there was Gypsy 7 doing its thing. Everybody else was long finished. But it was dragging up the rear. So we actually realized we had one mutant that was just slow. We never looked for slow mutants. Now, so what do you do with stuff like this? What you do is you clone them. You clone the gene that's defective. And I'm not going to go into a whole lot of detail about either cloning or what these genes are, but if you want me to, I could. All right? So most of my lab spends their time working on what are these genes. Now, I also do a lot of bioinformatics work and genomics level work. So we've done time points across this treatment, looking at what genes out of the entire genome of Arabidopsis. This is why we work with Arabidopsis. The genome is now fully sequenced, and we have all kinds of tools, molecular tools to use. So most of my lab is working on that. And we also did a time course throughout this cold treatment and afterwards, and we looked at the whole genome. So we got 432 genes selected that were differentially expressed between the treatment and wild type, or the treatment and the control. Now, what do you do with 400 and some odd genes? My students don't want to tell me I got that many. Would you want to work on 400 and some odd genes? No. You know, how many genes can any one student, undergrad, master's, or even a PhD student work on in their course of their time, right? But often when people do these things, they select some of the highest genes, the ones that are most differentially expressed. The problem with that is this is a signaling event. Signaling spikes on, it spikes off. It doesn't have to be the most, the biggest. So I didn't want to throw any of them out, which made everybody in the lab cry. Because they were really sure that I was going to make them do all of them, but I didn't. What we did was we took, whoops, wrong way, a bioinformatics approach. Let's go to here. So we took all of these genes, we did a database search to look at any genes that they might be connected to in any other situation, and we built a network. You guys spinning out the network? Come on, guys. I can kill time. So you look at this, and it probably doesn't mean much. There's a bunch of different colors. The different colors are the different time points. So the yellow ones were at 2 minutes. These were at 4 minutes. Red was at 10 minutes. And the blue ones were at 30 minutes. So these are the time points. Got to move a little faster, Sean. They got into listening instead of doing our trick. So this is our network, and you got to ask, what am I going to do with this? It's pretty, huh? What are you going to do with it? Well, what we're going to do with it is what I refer to as the Google Map model. When you're driving from Athens to Columbus, how many different routes are there? Which cities are you going to go through? So those of you that are now connected, this ball of thread that's going around is connecting you. Pull it tight, guys. Pull it up. Pull it tight. You guys, somebody back there, we got some? So the idea is, if I know that you are involved in gravity, I know it. I have a mutant, I know that you're involved, and I know that you're involved. If I pull this, who else do I get? I get all the linking people, right? So that's what we're going to do. They're still sweating the details in the lab. So this is what we're going to do. We're going to take this thread right here, and we're going to pull it and see who else is involved. Does that make sense? Because this is state of the art right now. This is where my field is after 12, 15 years. We have all of these characters. You notice the black box originally, that big blank? Well, now we have stuff in here. There are genes that we know are involved. These are the genes that you people represent. The problem is, if you look at this, it's not a black box anymore, so that's a good thing. But we don't have a clue how they interact with each other. So a colleague and I published this on a review as the gray cloud. It's not a black box, it's a gray cloud now. Okay? So this is what we're trying to have a more linear progression. What is step A, pull to get to step B? Just like pulling on the string. Does it make sense? We'll see how many. So what we're doing with this is we're actually going to get mutants, which are mostly available, for each one of you guys that are in between and characterize them. And then we'll start trying to connect the dots. Some of them, even though they're connected, there may be more than one way to get to somebody. I mean, if you look at these, you know, none of these are just connected in a straight line. There's all kinds of linkages. So we've got some sorting out yet to do. I got a whole career. Yes? We don't know which way is the most efficient. And the plant may not be using the most efficient way. So we're not looking for efficiency. We're just looking at now we have a possibility of being able to connect the dots. That's all. Okay? We have to be able to sort out this pathway some way or another. Yeah, seriously with the string. It makes your life harder. So this is what we can do on ground. This is what we can do here on Earth in my lab right now. These types of studies where we're looking at the genome and we're looking at basically a bioinformatics methodology, bioinformatics using informatics, computer technology to look at biology, to try to sort out signaling events in gene networks. That's the majority of what my lab does. Now, there are other ways to work on gravity. Remember that whole idea that there's no ultimate control? There's no gravity? Well, there are ways we can simulate microgravity. The real term should be microgravity because you're never completely without gravity. There's always something. You have them? One way, hang on just a sec. One way to do that is with a drop tower. Okay? So this is a kind of a classic way to do kind of a microgravity environment. The Glenn Space Center in Cleveland, they have a drop tower. Their drop tower is, I forget exactly. I've got a note here somewhere. Oh, there it is. So the Glenn drop tower is 79 feet. It's the 8th floor, so it gives you 8 floors. And when they drop things, it takes 2.2 seconds. Not a lot of time. Now, these are good for physics. Some physics experiments, those kinds of things, how fluids work in microgravity, that kind of stuff. It's really good for that. Now, the longest drop tower there is in Japan. The one in Japan goes down a mine shaft, and it is almost 500 meters. So that's 5 soccer fields, vertical. It lasts about 10 seconds. So we're going to show you a little demo here. I know you're all wondering what the ladder's for. Are you ready? Hopefully nobody gets wet. So we're going to have a little bitty drop tower. All right, turn it this, which one you give? I'm only taking one at a time. Okay. No, because I'm right-handed. I have a towel. All right, that wasn't so bad. So, can you turn that can, the other can? No. Just flatten them up. No, flatten them up. There you go, good enough. Okay, so as you probably figured out, there's a hole in the bottom of this can, under my thumb. Okay? Okay. What I'm going to do is I'm going to let the water out and hit that trash can, and then I'm going to drop the can. What you should notice is that the water quits pouring out of the can. All right? Did you see it? Did you watch the can instead? A lot of people do that. So we're going to do it one more time. A lot of people are following the can instead of, it's under here, right? Good job. That's okay. We'll get it afterwards. All right, you ready? Watch the water, not the can. See the water? Oh, this isn't going to hit right. Did you get it? So that's a drop tower. Very basic, very simple. That's a drop tower. And just to prove that there is actually still water in these cans. So that's a drop tower. That's one way to do it. Not very good for plant growth. Even at 10 seconds, I can't do a whole lot with that. All right? So the second possibility is called parabolic flight. I'm going to show us, maybe. I lost it. I had it on here. That's the one. So I'm going to show you a quick video. Parabolic flight, a plane. We'll let it go, then we'll talk. Come on.
La combustion physique des plasmas, des lasers.
We'll see if we can get a little further
Physique
into the
d'agrégation de
video.
poussière interstellaire, expériences avec des produits chimiques particuliers.
Here we go. Come on, guys.
Les deux sièges, les deux toboggans qui sont uniquement sur ces deux portes avant et les deux toboggans qui sont sur les deux portes arrière. Les deux portes du milieu ne sont pas utilisables.
Bienvenue à bord. Notre Airbus est rangé un véritable laboratoire de recherche volant, comme disait Stéphanie, dans lequel les chercheurs dans toutes les disciplines de biologie, physiologie, physique des fluides, des matériaux viennent effectuer de la recherche fondamentale, appliquée, parfois viennent tester des expériences avant de les embarquer dans des vaisseaux spatiaux.
So they're getting ready. What they're doing, the plane is going up at a very steep angle. In a minute, it's going to go over the top and head back down at just as steep an angle. A parabola.
L'injection à gravité martienne. Voilà, il ne reste plus qu'un tiers de la gravité terrestre. Ce sont des très légers sur Mars. C'est exactement la surface de Mars. On peut faire des
Allez, on est sur la planète Mars.
As you go over the top, you actually experience microgravity.
Voilà, on est sur la planète Mars.
See the guys hanging upside down? Now they just turned, and now they're coming back and they're going another round. So now they have gravity. You can see it going straight down. Now it's going to do this again. Every microgravity flight, they do about 40 parabolas. So I'll leave that there. So the idea of this, now that was the European one. The US, NASA has one. It's the KC-135, also nicknamed the vomit comet. Not everybody gets sick on it, but some people do. So as they come over, as they go up, and then you come over the top, you experience microgravity. Much like when you're on a really fast roller coaster and you go up and over, that's that same experience. All right, that same experience. So that gives you about 20 to 30 seconds. For those of you that saw the movie Apollo 13, and you saw the space, that they were in the space, I guess in the capsule, where they were floating around, that was all filmed in 20 to 30 second bursts on this plane. So that's how they got to microgravity. Does that make sense? It's kind of fun. Now, the way NASA uses it for the most part, there are experiments that you can do in that time window, but one of the things that they do for anything that's going to fly, that's going to go up, used to be in the shuttle, now in SpaceX, in the Dragon, when it goes up, they want to make sure it works and that the astronauts can actually do whatever it is they're supposed to do. There have been experiments where they needed to cross-pollinate plants. Can you actually do that? What are the tools that you need? So a lot of times they'll test these things on the KC-135 while it's under parabolic flight. Okay, that way you go up, there's a light that comes on, you know when it's going to hit, and then you try to see if you can manipulate whatever it is while you're in microgravity. And then, of course, you come back down. Now, the really cool thing about the KC-135, as I said, it usually goes about 40 rounds. So if you can get your experiments sorted out in 5 rounds, 10 rounds, you get the rest of them to play. And this is quite a bit of fun to play. So that's drop towers, and that's parabolic flight. Now, what I want to talk to you about is another way that we can do this, and this is particularly good for plant experiments. If you guys have noticed, I have several of these gizmos. This one, and there's one over there on that table. These are clinostats. Clinostats. Some of you, if you've been paying really close attention, you realize that they're turning. It's kind of hard to notice because they're turning really slowly. They're turning at about 1 rotation per minute. Maybe. Oh, there it goes. I moved this one time too many. Okay? So what I have in here, what you see in here is a plate of seeds. Some of you on your table have plates of seeds. And you can see the little bitty Arabidopsis seedlings. Okay, the little bitty Arabidopsis seedlings. So we plate them, and I really can't show you this because nobody can see it. If you can see the black line, we actually plate them along that line so that we stand them up and germinate them vertically. Now, what happens with the clinostat, if I want to do a gravity experiment, what the clinostat does is it simulates microgravity. Obviously, gravity's still here, but quite honestly, the plants are confused. Because the statists, remember the statists? They never settle down. They just keep spinning. Now, our research shows that yes, it knows there's a problem, and it knows that it's still spinning, but still it can't respond. So it's very symbolic of what actually happens in microgravity on spaceflight. All right? So yeah, if the statists, it's sort of like rolling down a hill really slowly. If the statists keep moving, you're never going to know which way gravity goes, right? So we can use these to actually look at genes that are involved in gravity as well. So this one rotates in this direction. Typically we put it in and they rotate. This one over here, if you can see it, you see that I've got plates strapped in 2 different directions. So we can actually play with the rotational plane. If a plant's growing like this, normally we rotate it this way, okay? So we would rotate it in this plane, okay? But on that one, I can also rotate it, this is going to make a mess, head over head. So I can rotate it this way depending on how I put the plate. Now, ultimately, the very best clinostat, the best type of clino-rotation is actually 3-dimensional. This one, if you really pay careful attention, it's rotating this way. I think you can see that pretty dramatically. But this is also rotating. So it's rotating in both directions at the same time, so the plants really have no orientation whatsoever. This one was actually, it's got a little hitch in its get-along, sorry. This one actually is made out of a bicycle frame, for those of you that are paying attention. It's made out of a bicycle frame, and we actually originally made this for a science fair project for a high school student. And then I've modified it several times to use it for other experiments. I had an undergrad that used it to look at magnets and the power of the strength of magnets and how magnets would affect plants. And of course, if you want to look at how magnets affect plants, one of the things you would really like to see is how that happens without gravity, because gravity is much more powerful. Gravity is more powerful than the response to light. So a lot of the experiments that actually fly on the shuttle and fly to the space station, many of them have been light, looking at light differences, because on the shuttle and in microgravity, you can actually see those responses much more carefully, things that are completely hidden here on Earth. So, of course, that leads us to the last bit. The last bit, flying an experiment. Flying an experiment is a great opportunity, actually. It's why my students are wearing NASA shirts at the moment. We will fly an experiment. We're going to fly an experiment probably in December or early next year. That experiment will actually just show it to me. We'll actually go in these. These are called bricks. NASA loves acronyms. Loves acronyms. So this is biological research in canister. So all it is a really heavy-duty canister with a screw top. And what we're going to do, I got to find them. I've lost them. Ah, there they are. What we're going to do is fly seedlings, like you see around, on little teeny tiny Petri plates down in those tubes. So we're going to take these Petri plates, we're going to put seedlings on them, just like the seedlings that you guys have that are sitting around. We're going to put them into the canister and then fly them. They're going to be shot up and stay on the space station, though. So they're going to be shot up, and then they're going to stay on the space station for 2 or 3 days, and then they're going to bring them back down to us. They will be seeds when they go up, and they will germinate in microgravity so that the plants themselves have never seen gravity. Then we're going to fix them, chemically fix them, and freeze them while they're still in microgravity so that they don't experience coming down. And then what we're going to do with it is we're actually going to look at genome and proteome. We're going to look at all the proteins and all the genes that are differentially expressed between these guys that went to space and those guys that stayed on the ground under the exact same conditions. Because we're going to build another network. Because we want to actually expand our network analysis to actually look at what and intersect those networks to see what is truly gravity related versus what might be some other effect. That's all I have for you guys today. Yes, questions? Yes, it will all be in dark. So the question is, aren't they all going to be in the dark, and wouldn't I expect others? I will have a ground control that's exactly the same, so we should be able to tease out those things that are just dark or light. This is one of the things about space flight. You have very limited space, literally limited space on the ship, and you also have very limited time. So the proposal that I wrote was designed to go up in 2 of those canisters and to be up for 3 or 4 days because that's what they had to offer. There are other setups that you can use, but only 1 or 2 experiments actually get selected at any round. They've actually given us 2 additional canisters now, so we'll have 4 canisters that go up. So we hope to be able to tease this apart. We have lots of steps left. We're currently in the phase called definition. So we're trying to define our experimental parameters and make sure that they're going to work perfectly in there. I mean, this is a lot of money, shooting these seedlings into space. So we want to be sure that our data are good when they come down. So definition is our first stage, and then there's a couple more stages before we actually get to fly, which is why it's going to take until December or next year for us to go up. Yes? The question was, if gravity affects genes in plants, does it affect genes in the humans? Yes, absolutely. There are definite effects of spaceflight on humans. You can see that there's a loss of bone density. When they're in space, there's a lot of fluid buildup in the upper part of the body because without gravity, it doesn't drain down. So there's a lot of different effects that occur, and that's one of the things that NASA is spending a lot of money studying, with the idea of if we're going to send people into space for any long time, we actually need to know what kind of effects they're going to have. No? Anybody else? Yep. Yeah, she asked me when the flight goes up. Of course, when the flight goes up, I mean, basically it's a rocket, and it launches pretty hard and fast. But my plates will be seeds. The plants won't have germinated yet. And over the years, that's what we found has been the best, to send them up as dormant seeds, and then they'll germinate in space. So the plants themselves will never see it. Does that make sense? The seeds will hit. I'll get you next. Go ahead.
Instead of going to microgravity, put your seedlings in a centrifuge and drive the gravity very high and see what the effects will be. I mean, it's the same idea.
Right.
Going from very low to very high.
Right.
But rather than going from very low to zero, simply go the other way. It would be simple to have a centrifuge. You could run it for hours.
And we do those. Those experiments do occur. It's just that this is the other side. So, I mean, all that research does go on, and NASA does have centrifuges that will do those things, and a lot of schools have them as well, kind of these big rotators. Yes?
Yeah, like, what would be like the sort of practical implications for like that gene and what you're doing?
The practical implications of gravity, kind of as I started off today, you know, we have evolved with gravity. So everything that is here has been affected by gravity and responds to gravity. So there are potential crop improvements that we could do, horticultural improvements that we can do. There are still some plants that have these sort of funky little things. So as far as growing additional food or growing it in different habitats or different places, and of course NASA's interest is what happens if they send them into space. You actually can't just pack a huge suitcase and send them with everything they need. I mean, we have a huge bioregenerative life support experiment going on here on Earth. We are in tight symbiotic relationship with plants. Without plants, we wouldn't be here. And so if we're going to do any long-term, whether it's a space station or another planet or whatever, then we have to know what's going to happen to plants. For me personally, this is an incredibly exciting, interesting biological question. This is a fundamental question for how plants grow, one that we still don't know very much about. So that's my real interest in it, just the fundamental biology and the knowledge that we can gain about how plants work. Yes?
Yeah, I was curious how you handle growth media and watering, just keeping the plants.
You really can't water anything. These will be on auger, so they'll have everything they need. These will be, auger is sort of like Jell-O. It's kind of a Jell-O mix. So they'll have all the nutrients and water that they need for the short time that they're going to be there.
So they're rooted in the auger?
They're rooted in the auger. And the auger has sucrose in it, so we're feeding them in essence, so they don't actually need to have light to photosynthesize. So we're going to feed them. Anything else? Other questions? Then I'm going to end where I started. Whoop.
From online, are you just sending the wild ones into space?
Yes. We are just going to send wild type because this is a foundational. Nobody's looked at the proteome, the proteins yet. There's a lot of things that go on with RNA, transcripts, and we know that. But not all transcripts. Once DNA is made to RNA, it doesn't necessarily always go on to be protein. And even when it does go on to be protein, sometimes it's inactive protein. So it needs to be modified in some way, post-translational modification. So we're really interested in the proteins because they are really the end products of gene expression, and they're the workhorses. They're the ones doing all the work. So it's the proteins that we're interested in, and this will be the first time that anybody's looked at the proteins in space flight. So we need a baseline. Again, we have 4 canisters. We need a baseline so that we can start looking at other potential experiments later. Yes? There actually has. There has been a considerable amount. He asked about how much kind of plant research has gone on, and there's actually been quite a bit. And the nice part about plants is you can have enough plants go to space that you have enough replicates to have statistical validation. Tell Gar not to call me. That's a colleague who's in Oregon who wanted to he's probably watching the live feed. Huh? Okay, so he's watching the feed. So where was I? Oh, plant experiments. Yes, there's a lot of plant experiments that have gone into space. Like I said, you can have enough replicas. Whereas with a lot of the human experiments or the mice and stuff, you could only take 2 or 3. Or you could only, I mean, even in the shuttle days, there were, 7 or 8 astronauts, and that was kind of it. So the replicates are difficult. What kinds of things have they looked at? The initial thing was, could you even grow a plant in space? I mean, germination, that fundamental piece of plant biology, requires gravity. So what happens to a plant if you take a seed to space? Will it actually germinate? And it does. And the roots go whichever way they want, and the shoots go, and they wind up to be a tangled up mess. Now, subsequent experiments, you have to realize, spaceflight, we do kind of one step at a time. Subsequent experiments have actually shown that you can orchestrate which way they go. If you have a light and water, roots will go to water in the absence of gravity, and the shoots will go towards the light. It's not quite as straight a plant, if you like, but it still has an orientation. Then, of course, there were the experiments that actually proved a plant could go seed to seed, and that you could recover seeds in space, and that those seeds were, again, viable, and that they would germinate themselves. So these may seem like very basic questions, but in a totally new environment, these are questions that had to be answered. So those are some of the questions that have gone up. Okay, so with that, I think we are adjourned. I'm happy to stay up here and answer questions, or you're welcome to come up here and look at everything, play around with it, whatever you like. Thank you, guys. I appreciate it.